Hi, my name's Paul Frandsen. I'm an assistant professor at Brigham Young University in Utah. And today I'm going to be speaking a little bit about some of the work we've been doing to understand the genomic basis of insect silk. So the title of my talk is Hanging by a Thread, Using High Fidelity Long Read Sequencing to Uncover the Genomic Basis of Insect Silk. And I'd first like to thank my colleagues. I've worked with many people on the various projects that I'll be describing in this presentation. And some of them are listed here as co-authors in the presentation, but then others will be acknowledged at the end. And so first, I thought it would be useful just to talk a little bit about silk. So silk is a single name that we give to actually multiple kind of types of materials that share some common characteristics. So it's not necessarily that each silk is governed by the exact same genes, for example, but that their phenotype or what we observe in nature is very similar. And so essentially what it is a proteinaceous substance that is eventually spun into fibers. And so this proteinaceous substance has a genomic basis. There are genes that encode for the proteins that end up being spun into these silky fibers. And silk is often flexible, it's often sticky, and it's used across multiple lineages of arthropods. So spiders use silk, moths use silk, as we know from their cocoons. I'll be talking about a special group of insects called caddisflies that use silk, but it's actually used all across insects. There's grasshopper relatives that use silk. There are wasps and bees that use silk, etc. So in total, more than 800,000 species of arthropod or use some sort of silk. And silk is a very useful biomaterial, and it's been used for centuries. And primarily, the products that we talk about from silk are derived from a single species called Bombyx mori, or the silkworm moth. And so the silk industry or the sericulture industry is largely based on rearing these silkworm moths and then harvesting silk fibers from the cocoons that have been reared, as you can see in these images here. And so while silk is a very useful biomaterial that's used for all sorts of different things, including textiles, etc., it's kind of interesting that we primarily only use silk from one species when there are, 799,999 other species that also spin silk. And so my research is kind of focused on looking at other species that spin silk and that have kind of unique material properties that might be advantageous to silk spun from the most commonly used species, such as Bombyx mori, this silkworm moth. And so the group that I'm most interested in is this order of insects that we call caddisflies. The scientific name of caddisflies is Trichoptera, meaning hairy-winged. And there are about 16,000 named species of caddisflies. So that's a lot of species. That makes them the second largest freshwater radiation on the planet. So they're a very diverse group of insects that live in the water. So the larval forms live in the water. And if you look at this diagram on your right, this is a phylogenetic tree, or basically an evolutionary tree that shows the evolution of this group of insects. And on the right-hand side, you'll see many different little illustrations of the different ways that caddisflies use silk. And so they've been sometimes referred to as nature's underwater architects. And this is because they use this special adhesive silk to build different structures. And caddisfly silk is really interesting, and we'll talk a little bit about this more. But I'm really interested in the evolution of caddisflies, and so that's why I became interested in silk, is because if you want to understand why caddisflies have been successful, you have to think about their silk. In addition, I have colleagues who are bioengineers who are very interested in applications of their silk. The silk is sticky underwater, it's very strong, and it has some desirable properties that make it a good kind of model for a biomaterial. And so they're interested in develop biomaterials based on the things that we discover in caddisfly silk that could eventually be used in things like surgical adhesives and so on. So this is a larval caddisfly, larvae meaning the immature stage of the insect. And this is a micro CT scan. So basically, a bunch of X-rays were taken to reconstruct this 3D image. And what we've highlighted here is the silk gland. And what you'll notice is that there are two silk glands, and they're actually derived from modified salivary glands. And so the silk glands are pretty large. And whenever you see a large organ in an insect, you know that that's taken a lot of energy to generate that organ. And so therefore is probably important to the success of the species. And so a lot of energy is put into generating these silk glands, but also generating the silk. And what you'll notice is there are two silk glands, and then they kind of come together in a single spinneret at the mouth parts or of the organism. And so what these caddisflies do is they spit out silk, and they kind of use it as a tape to tape these different structures together. And it's sticky underwater, or it adheres to things underwater. It's a really cool biomaterial. And here are just some of the things that they make with their silk. You can see there are some caddisflies or caddisworms that are free living, but there are others that make little cases, these little tube cases, as you can see in the illustration under G, where they take pieces of material from the stream, whether it be rocks or sediment or plant material, and they use this underwater adhesive tape to tape it together. And then they put this around themselves and walk around on the bottom of the stream like a hermit crab, and they're able to use that for camouflage, for protection, and they even use it for respiration. So these little silken homes have allowed caddisflies to live in poorly oxygenated waters, like slow-moving waters, lentic environments, where they're able to kind of move their abdomens, undulate their abdomens, and bring water in through their cases to oxygenate their bodies. But they also make really a bunch of other interesting things. If you look at H or I, these are little underwater homes that some caddisflies have made. And they've made them entirely of material that's on the bottom of the stream bed and silk. So in H, you can see it's a pretty sophisticated filtration system that brings water and food in through the caddisfly larva. And then I, they've made this really fascinating home where the entire thing is lined with silk, complete with a little web. You can see the web down by the head of the caddisfly, and water is kind of filtered through that web, and it allows the caddisfly to capture prey and eat things off of the web that is captured. And so they use the silk to great advantage. There's just some images of caddisflies walking around. These are the tube case caddisflies, those that make a portable case. And artists has even discovered that you could take caddisflies out of their natural environment and lay them in a bed of gold, and they can make art. And so this is a pretty fascinating use of silk by the caddisflies. And what you'll notice here is that this caddisfly has taken this really kind of, size selected this little piece of gold and is trying to find a place to add it to its case. And we also notice is it has its claws that it's holding this piece of gold with, and then it's basically spitting out this adhesive tape onto the gold and placing that into its case. And this is how most of those tube casers work, is they spit out this liquid silk that hardens in the water and adheres to a surface and allows them to make these structures. Here are some images of caddisflies that make fixed retreats. So instead of making a tube case that they walk around in, they make a little home that they live in on the bottom of a rock. And what you'll notice is that there's a web, kind of like a spider web, and that's entirely made of silken threads and allows them to capture prey. Kind of even more interestingly, in many streams, these caddisflies are so abundant that these silken homes that they've made stabilize the stream bed and prevent erosion. So they're very important ecosystem engineers. Colleague of mine, Russell Stewart, who's a co-author on this talk, has done a lot of work on the properties of caddisfly silk and comparing it to other biomaterials. So what you'll notice is that kind of the further up and the further to the right that you get, the stronger the silk is, or the material is. And you'll see caddisworm silk here in this big orange circle labeled K. is stronger than the human meniscus, for example. So it makes a really good candidate for a biomaterial that we might want to mimic. But all of this exciting stuff about silk, but how do we get there? How do we understand how silk is produced? And what I'm really interested in is understanding how variation in fibroin genes affect phenotypes. So silk in caddisflies is derived from basically two types of fibroin genes. So the first one is the heavy chain fibroin, and the second one is the light chain fibroin. So heavy chain fibroin, we think, forms the structure of the silk, while light chain fibroin kind of holds the silk together. And the first step towards figuring out what these genes do is to measure the variation among those genes. And so we've done some work on this. And some recent work that we've published was looking at the conservation of the structures of light chain fibroins first. These are kind of the simpler protein encoding genes that we looked at. And what you'll notice on the left here, the top line where it says BM is Bombyx mori. So that is the standard silkworm moth that we get derive most of our silk from. And then the HO is a species of caddisfly, Hesperophylax occidentalis. And this is an alignment. It's actually derived from a much larger alignment that included many more species. But what you'll notice is there aren't very many conserved amino acids. So what you would expect if these two species were the genes were very closely related, they would have many conserved positions. But if you look across the gene, those highlighted in green are conserved amino acids. And so there aren't very many conserved amino acids. However, even despite the lack of conservation of amino acids in this gene, the structures are very conserved. And so we folded all of these up with AlphaFold. This was work led by my colleague Russell Stewart at the University of Utah. And we could compare the three-dimensional structures of these genes across the different orders. And we noted that there is a high level of conservation. So if you look at the diagram on the bottom left, there are three images, A, B, and C. A is the caddisfly, B is Bombyx mori, and then C is those structures overlapped. And where it's colored is where the structures differ a little bit. And what you'll notice is that those structures, despite low levels of amino acid conservation, have very strong structural conservation. And so perhaps the light chain fibroin is not the best place to go to understand why caddisfly silk has differing properties from moth silk, for example. And so we decided to go to the next gene, which is the heavy fibroin chain. And the first heavy fibroin chain sequences were published by Yonemura et al. For caddisflies in 2009. And this is a little bit of a complicated diagram, but on the left here, there's A and B. A is the N terminus of the gene, and this is, again, an amino acid alignment where the first two sequences on that amino acid alignment are caddisflies, and then the next three are moths. And then in B, this is the other end of the gene, the C terminus, with the same taxa or the same species. And you'll notice that A ends in ... You start to see some repetitive region that ends in ... And then B again begins with ... The reason for this is that we were unable in 2009, the sequencing technology was not well equipped for sequencing and assembling these complicated proteins. This was followed up with work by my colleagues at University of Utah, where again, they were unable to sequence and assemble through the entire gene. And this is because this gene has two conserved regions on the ends of the gene, but the middle is totally repetitive. So this is just a little diagram that shows, it's a cartoon that basically shows how this gene is, the structure of this gene. So if you see these little blue ends, those ends are the termini that are conserved, and they're non-repetitive. But internally, there are these little blocks, and those blocks are repeated over and over again throughout the length of the gene. And early high-throughput sequencing technologies generated very short fragments of DNA. So basically, you take your chromosomes, you cut up the DNA, and then you sequence these little short fragments. into what we call sequencing reads. And then you take those sequencing reads and you try to place them back together with overlaps to try to reconstruct what the genome would have looked like. And with a gene like this, you can imagine these little green lines. Those are sequencing reads, short sequencing reads. And what you'll notice is that these sequencing reads are shorter than repetitive regions. And so when you try to reassemble the repetitive regions with a bunch of little reads that are shorter, it's like an impossible puzzle. And so you're unable to assemble it. However, more recently, long read sequencing has come onto the scene and generates reads like these yellow reads that are much longer, that can span the entire repetitive region. And so with these, you're able to reconstruct the entire gene region. And this is really important for caddisflies, we think. And so we published a couple of papers with the first full-length heavy fibroin sequences. First in this caddisfly Stenopsyche tianmushinensis, and then in this caddisfly Parapsyche elsis. And we were able to sequence through for the first time the entire repetitive region. And so why is that important? Why do we care about what the repeats look like? Well, previous work has shown with tandem mass spec that there are some interesting peptides in the repetitive region. And these peptides include some modifications to some amino acids that we call phosphorylation. So what you'll notice here, just if you look on the right-hand side where the peptide sequences are, wherever there's an S, that's the amino acid serine. Wherever it is underlined, it means that it's been phosphorylated, or there's a phosphate group on that. And if you look at this diagram of the entire gene, this is a little bit complicated, but on the top, there's the conserved N terminus. On the bottom, there's the conserved C terminus. In the middle, these are the repeats. So 1A corresponds with 1A in the diagram on the right. And then, for example, you'll see something like 3B, which is repeated over and over again throughout the plot on the right. And this is kind of a diagram of the gene that shows how these repeats are repeated over and over again. And we think this is really important because these repeats form the structure of this particular protein. And the way that it does that is that these serines have phosphate groups, which is illustrated here in this diagram. You can see that phosphate groups, and those phosphate groups bind with calcium ions or some sort of other metal cation, and that generates the structure of the silk. And so the more phosphate groups there are, or the more densely packed those phosphate groups are, the more bonds it will create with those metal ions, granting strength to caddisfly silk. And this is a unique adaptation to caddisflies that is not known to occur in moths, for example. And this is part of the reason that caddisfly silk is so strong. And so you can actually measure the strength of a single fiber, for example, by attaching them to calipers and then pulling them apart. And as you pull apart that fiber, it breaks those calcium phosphate bonds, but allows the fiber to extend. And the fiber is viscoelastic, meaning that it actually regains its form after some time. So you can expand it out and then bring the calipers back together. And eventually, over an hour or so, the caddisfly silk will regain its original length. And so it's a pretty cool fiber for biomedical applications. We got really excited about using these long read sequences to try to generate caddisfly genomes. And so we kept trying to do it. The first two were with an older version of PacBio technology and then an older version of Oxford nanopore technology. So we decided to try to sequence as many of these as we could to try and understand the diversity of these genes across caddisflies. And we were first using Oxford nanopore technology, one of their older technologies, the 9.4.1 pores. And we started to notice something interesting. So this is a little bit of a complicated diagram, but I'll try and walk you through it. On the top, there is a sequence, and this is the consensus sequence of the heavy fibroin chain protein. So what we noticed is that we were getting really poor assemblies with Oxford nanopore, the older Oxford nanopore tech. And the reason for this is because it is noisy data. So basically, even though they can generate long reads, there are many errors within those reads. And so each of these lines represents a read. And that read has been kind of mapped back against the consensus sequence of the protein. And anywhere you see a black line within a read, that is a mismatch. And so you'll notice there are many mismatches of the reads compared to the consensus. And so we were unable to resolve this region. Even though we got assembly, we were unable to find exactly where the gene started and stopped, and we were unable to reconstruct that repetitive region. And so pretty soon after that, our institution got one of the newer PacBio instruments. Since then, there's a newer one that we're getting, but where they're able to generate what we call hi-fi reads. These are reads in which the individual molecules still are read. They become a little bit noisy, but basically the technology allows you to read a single molecule multiple times. And by reading that single molecule multiple times, you can self-correct the molecule and you end up with a very highly accurate read. And so we were excited about this technology to apply to our silk proteins to try and kind of understand the genetic basis of silk. And this is, again, a very similar diagram to what I showed you before. And you'll notice that many of the reads actually match much better to the consensus. And so this was kind of surprising to us. We were very excited when we got these new data back. And we noticed like, okay, like this looks a lot better. But if you kind of relax your eyes, you'll notice that about half of those reads don't match very well at all. But half of them actually match the consensus well. And so when I first saw this, I realized, oh, caddisflies are diploid. They get a copy of chromosomes from their mom and a copy of chromosomes from their dad. It's possible that there's a lot of heterozygosity here. So I wrote a quick script that when I first got these data back, which was right after the technology was released, and binned these reads into those that had mismatches versus those that had fewer mismatches. And then I generated 2 new assemblies, and voila. You can notice that the reads match back to the consensus with much higher accuracy. What you'll also notice is that this particular gene is 26,000 base pairs. And then we took the reads that had a lot of mismatches, reassembled them and mapped them back. And suddenly that particular allele of variant of that gene was only 20,000 base pairs. So there's huge length variation between haplotypes within this particular gene region. And so this was the first time that we've noticed that there was a large amount of heterozygosity. And this is kind of important for understanding this gene because we're trying to link the properties of this gene exactly with its phenotype. And to do this appropriately, we need to really understand the variation in the protein. And so these are just schematics that show the repetitive regions of the gene repeated over and over again. You can see that one allele is much shorter than the other. And so why is this important? Well, in one of the caddisflies that we're interested in, this net spinning caddisfly, we're very interested in understanding how these genes fold and how that provides structure to the protein. And so you can take this set of repeats, all these repeats that are outlined in red, and then you can generate predictions using something like AlphaFold to assemble this into a beta sheet protein. And then we can kind of understand how frequently these beta sheets show up in the gene itself, and then potentially eventually make some prediction for what the phenotype of this particular variant would look like, or this particular allele of the gene. And what you'll notice, even between these two alleles, the density of this particular repeat really varies. And so this probably has an effect on the phenotype. And so how are caddisflies using this variation to their advantage? Maybe using one allele to create one strength of silk and another allele to create another strength of silk. This is the type of thing that we're investigating now. And so we were really excited that we noticed these patterns in caddisflies. This is something that had never been discovered before, to allow us to understand that there's much more variation. We call it hidden variation in these gene sequences. And so we decided, okay, let's look at other caddisflies. So we sequenced more caddisflies, and this is kind of a simplified diagram where it shows these ribbons, these blue ribbons basically show where the gene is conserved between alleles. And then whenever there's white, that represents some sort of insertion or deletion in one allele opposed to the other. And we noticed that there was a persistent pattern in every single caddisfly that we sequenced. This heavy fibrin silk gene had substantial allelic diversity. And this is really important for us to be able to understand how to make connections between the genotype and the phenotype that will eventually lead to translational applications of the silk. And since we noticed this in caddisflies, we decided that we would look at other insects and arthropods. And we first started with a butterfly. This is Vanessa cardoi, a pretty common butterfly. You've probably seen them, the painted lady butterfly. And again, we pulled out this silk protein has been under evolution for almost 300 million years. And even though there's very little in common with the repeat structure itself, the patterns of variation are very similar, with these large insertions and deletions between alleles and large length variation. But maybe this isn't surprising, given that this is a gene that shares an evolutionary history. So caddisflies and butterflies and moths, the ancestor of that whole group of organisms must have had some prototypical heavy fibrin chain protein, because they share that, and we can establish homology. But what about other organisms that also spin silk? Would we find anything about those other organisms that share the same pattern? And so we decided to look at a spider. The spider we chose is Argiope argentata, and it actually has many proteins that many genes that encode for proteins that generate different types of silk. So it uses silk, many different types of silk, to make it its web and do other things. And so we just looked at 2 of those particular genes to evaluate the variation that we'd see, and see if there's any sort of pattern that would suggest common mechanisms or convergent evolution, because this silk is totally independently derived. It doesn't share an evolutionary history with the caddisfly and butterfly silk. And what we found was really some strikingly similar patterns, that even though these proteins are really different, they have the exact same pattern or very similar patterns of genetic variation. And so this is super exciting for us, because we kind of understood that there must be common mechanisms responsible for the molecular evolution of these really important structural proteins. And so now we're following this up with other research to see how that varies across other structural proteins or other branches of the tree of life. And this paper was recently published earlier this year. And so I'll just kind of finish up this particular section to just show what the effect high fidelity long read sequencing is having on biodiversity genomics, or those of us who study genomics that in non-model systems, not in humans, not in Drosophila melanogaster, but in things that had never had their genome sequenced before. This is just a figure from one of our recent publications that shows that these genomes that are assembled with high fidelity long read sequences are much better. If you look at the bottom, I and J, one of the metrics we use to evaluate how good assemblies are is this metric called contigen 50. Basically, it tells you how put together the genome is, and the purple represents these high fidelity long read sequences, and you'll notice that they're much better than other existing technologies, either noisy long read sequences or even short read sequences. And so it's a very exciting time to be someone who sequences these types of animals that are not well known, but we have the tools to be able to do it really well and to unlock all of these secrets of biodiversity. And we're starting with silk, but we're excited to look at other elements of biodiversity and how that can drive our understanding of genome evolution. And so in conclusion, long read sequencing technologies, finally, we can resolve these heavily repetitive silk genes. We've been trying to do it for decades, but we're at this exciting time where we can generate high quality assemblies. And long and high quality sequencing, like PacBio Hi-Fi, for example, is especially important when there's substantial heterozygosity. So we were unable to resolve these genes with long read sequencing, even that was noisy. But when you have a really low error rate and long reads, you're able to finally generate these assemblies. And because of that, we were able to unlock this hidden variation that we hadn't been able to find before. And lastly, there's just massive allelic variation in silk proteins. So these differences are probably really important for phenotypes. And so we really need to understand the variation and properly characterize it when we're characterizing the phenotypes. And I think there's also something common about these mechanisms that generate the variation that we're observing in these in these big structural repetitive proteins. And we're observing it in silk, but we've also been able to observe it in other large repetitive structural proteins that we've looked at so far, which is really helping us get more information about how the evolutionary process works, how evolution works on large structural proteins, and kind of how those are the origin of those proteins, but then also how they're maintained. And so I think continuing research in this area is going to be really important. With that, I would just like to acknowledge all of the different organizations and funding sources and collaborators and students that have contributed to this research. And I would like to thank you for listening. And I'm always happy to field any questions. Feel free to reach out. So thank you.